Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A Biology Unit 1 for January 2021. This is the Part 2 video. I'll put the link to the Part 1 video below the description box. Let's begin with question 5. Question 5 says the structure and properties of the cell membrane are important in controlling which molecules can enter and leave a cell. The photograph shows part of a cell membrane as seen using an electron microscope. So we can see this part, and they showed us the cell membrane here. So they say the width of this cell membrane is 5 nanometers. A phosphate head of a phospholipid is about 0.8 nanometers in diameter, and the fatty acid tails are about 1.25 to 1.75 nanometers long. They want us to explain how this electron micrograph provides evidence for the structure of the cell membrane, and they want us to use the information given to support the answer. So I began by drawing something here so that we can visualize as we add up. They told us the phosphate, which is the head here, has between 0.8 to 0.9, so I put it there. And then if this tail is 1.25 to 1.75, if you add 0.8 to that, we can see we get a 2.05, and that to that, we get a 2.65 as shown here. So that is going to be what I'm going to use in my explanation. I said the micrograph shows that the membrane is made up of 2 layers. I think you can see there are 2 layers, the lower one and the higher one. So it's made up of 2 layers, meaning it is a bilayer. Also, the micrograph can help us to measure the size of the phospholipid to be between 2.05 nanometers to 2.65 nanometers. I think you can see how I got this is by adding the lowest value here to the lowest and the highest value here to the highest. I also showed you here how I get the 2 values. They have told us that the width is 5 nanometers. When we double this to create that bilayer, it's going to be between 4.1 to 5.3. That should be around 5 nanometers. So I said the membrane, which is a bilayer, is 5 nanometers from the micrograph, and this is in range of 4.1 nanometers to 5.3 nanometers. That is in agreement with what we see from the micrograph. So if you had written these 3 statements, you don't need to draw this. You'll be able to get 3 marks. Let's continue to the next part. Here they say cell membranes contain cholesterol. The molecular mass of cholesterol is carbon 27, hydrogen 46, oxygen. The diagram shows a cholesterol molecule. So here we have a hydrocarbon tail, we have hydrocarbon rings, and we have the whole thing here. So they say explain the location of cholesterol in the cell membranes. They want you to use the information in the diagram to support your answer. So if this is a cholesterol molecule, we already know since these are hydrocarbon rings, they're going to be hydrophobic, and these are going to interact with the phospholipid tails, while this part that has OH, this part is going to be polar, it's going to interact with the phosphate, which is charged. So I say cholesterol is located within the fatty acid tails of the membrane. This is a fact. Also, cholesterol is nonpolar due to its hydrophobic rings. We can see these hydrophobic rings, they are nonpolar. So because these are nonpolar, they will interact with the fatty acid tails. So the fatty acid tails are also nonpolar. So the nonpolar part of the cholesterol and the nonpolar tails will interact. So the OH group on the cholesterol will be oriented towards the phosphate head of the phospholipid. This is explaining the location of cholesterol in the cell membrane. And again, to emphasize, these are nonpolar. They will interact with the nonpolar part of the phospholipid, which is going to be the fatty acid tails. This part here can interact with the phosphate, so it's going to be oriented towards that part. Let us continue. Part C says, the diagram shows the permeability of cell membranes to some chemicals. Here we have sodium and chloride ions, we have glucose, we have steroid, and water. This is increasing permeability. They say the sodium and chloride ion, glucose, and water are polar chemicals. A steroid is a non-polar chemical. They want us to describe the dipolar nature of water. So if I try to draw the structure of water like this, for chemistry students, you know that oxygen is more electronegative than hydrogen, so oxygen is going to get a partial negative charge, while the hydrogens are going to get a partial positive charge. Because of this, we can see polarity has been created. So here I said, in a water molecule, there is a partial positive charge on the hydrogen and a partial negative charge on the oxygen. So this describes the dipolar nature of water. Moving on. Here they say explain the permeability of cell membranes to each chemical shown in the diagram. I began with sodium and chloride because these are ions, they are ionic, so they pass through the channel proteins, or you could say carrier proteins, by facilitated diffusion, or it could be by active transport depending on the concentration gradient. Also, when we go to glucose, glucose is a polar molecule, so it passes through channel proteins by facilitated diffusion. Since steroids are nonpolar, a steroid can pass directly through the hydrophobic tails by diffusion, or what people call simple diffusion. And lastly, water is polar, so it moves into the membrane through channel proteins, which are called aquaporins. Water will not directly go through the membrane because it's going to be polar. It will not be able to go through the hydrophobic tails. So this brings us to the end of question 5. Let us continue to question 6. Question 6. Gas exchange surfaces have specific adaptations. Lungs contain the gas exchange surfaces of humans. Smoking causes lung damage. The graph shows that there is a correlation between smoking and death rate from lung cancer in men. So the vertical axis has the number of cigarettes smoked per year, and the horizontal axis has the different number of years. We can see the cigarettes smoked, and we can see the death rate. Of course, we can see as the number of cigarettes smoked increase, we see there is an increase in death rate, and when those go down, there is a decrease in the death rate, although there is a lag in between the two. The first question says, explain how this graph shows that there is a correlation between smoking and death rate from lung cancer in men. And again, to emphasize here, they are looking for a correlation, not a causation. It's a correlation. So I said, both the lines for cigarettes smoked and those for death rate rise and fall parallel to each other. This shows a correlation. However, death from lung cancer has a time lag after the line for cigarettes smoked. So it means as this one increases, there is a time lag. It does not immediately begin. There is a time lag. Also, as this one goes down, there is a time lag before that goes down as well. So there is a time lag in between the two. But we can see as one increases, the other increases. As one decreases, the other decreases. But there is a time lag before one reacts to what has happened from the other side. So correlation, not causation. Moving on. Smoking is a cause of emphysema. Emphysema reduces the surface area of the alveoli. So they say people with emphysema have weakened alveoli that can collapse, creating fewer but larger alveoli. Explain how this will affect gas exchange in people with emphysema. I've already said this reduces the surface area, and if that occurs, it means less oxygen is going to be taken in, or less gaseous exchange will occur, less oxygen will diffuse in, and less CO2 will diffuse out. So I said, because the alveoli collapse onto each other among people with emphysema, this leads to a smaller surface area to volume ratio and a lower rate of diffusion of oxygen into the bloodstream. The next part says, brood embryos develop inside hard-shelled eggs. Gas exchange occurs across the shell of the egg. The oxygen diffuses into the bloodstream of the developing embryo, and carbon dioxide diffuses back out. The diagram shows a section through the shell of an egg. So this is a section we see there are pores through which these gases are going to pass. We have a hard impermeable shell. That is for protection. Down there are membrane linings inside the shell. The thickness of the egg shell is 0.5 millimeters, and the density of the pores in the shell varies from 40 to 400 per centimeter squared. They want us to explain the factors that will determine the rate of diffusion of gases between the air and the tissues of the embryo, and they want us to use the information in the diagram, the question, and our own knowledge to support the answer. So here I began off by introducing the theory behind diffusion and what factors increase the rate of diffusion. So I said, based on fixed law of diffusion, a higher rate of diffusion depends on, number one, a large surface area to volume ratio. shorter diffusion distance, and greater concentration gradient. A shorter diffusion distance ensures that the embryo receives the gases, in this case oxygen, in a shorter period of time. Also, diffusion depends on temperature. Of course, this is very important. The higher the temperature, the higher the kinetic energy possessed by the gases, and the faster the diffusion, or the faster they diffuse. When we look at the structure given to us on the previous page, I also say that based on the structure of the egg, a higher rate of diffusion can be obtained if there are more pores, and if these pores are wider or they have a wider diameter, a faster rate of using up of oxygen by the embryo. So if the embryo is using up oxygen efficiently, then there is going to be a good concentration gradient, therefore there will be faster rate of diffusion. Faster blood flow through the embryo, and if there is a thinner thickness of the membranes lining the inner walls of the shell, and a higher body temperature of the embryo. And again, this is going back to a higher temperature, higher rate of diffusion. So finally down here I say the rate of diffusion also depends on the diffusion of the oxygen in the medium it passes through. This should be passes through. The medium it passes through. For example, if it passes through air, we need to know the diffusion coefficient of oxygen in air and see if it favors a higher rate or faster rate of diffusion or if it does not. If the diffusion coefficient of oxygen as it goes through the air favors a higher rate of diffusion, then it's possible for diffusion to be faster. So this brings us to the end of question 6. Let us continue to question 7. Question 7. Several factors increase the risk of cardiovascular disease, CVD. The diagram shows some factors that increase the risk of CVD. Complete the diagram with one lifestyle risk factor and one non-lifestyle risk factor. If we look at lifestyle risk factors, there is smoking. Of course, they've given us inactivity, they've given us high blood pressure. I included smoking. If somebody has high cholesterol intake or high salt intake, all this can be lifestyle risk factors. These depend on how your lifestyle is. However, when we go to the non-lifestyle factors, there is genetics. You cannot change your DNA. There is race or ethnicity. You cannot change who you are. There is gender. You cannot change if you're female or male. And there is something else to do with age. You cannot change your age. So these could be the non-lifestyle factors. Let's continue. The next part says the graph shows the effect of exercise on the relative risk of death from CVD. So vertically we can see the relative risk of death from CVD, and horizontally we can see energy needed for exercise. We have light exercise, moderate exercise, and vigorous exercise. For those who do light exercise, no matter what, there is a relatively higher risk of death from CVD in comparison to those who do vigorous exercise. So for vigorous exercise, it's always lower in comparison to all the others for all energy requirements. So the question here says, describe the conclusion that can be made from the information shown in this graph. I can see from this graph that overall, increase in the intensity of exercise leads to a decrease in the relative risk of death from CVD. So with more intense exercise, we see there is always a lower risk in comparison to the others that are not doing very intensive exercise. I also said for all intensities of exercise, increase in the amount of energy needed for exercise leads to a decrease in the relative risk of death from CVD. To show this, I could choose to draw a vertical line. At the same amount of energy required, something like that, if I draw a vertical line here, we could also see what values we're going to get on this side for all those three curves. So we can see that for this value here, for those who do light exercise, there is a higher rate, a higher relative risk of death from CVD in comparison to those who do moderate and in comparison to those who do vigorous at the same energy requirement. So let's continue. Here they say dietary antioxidants may reduce the risk of CVD. Explain how dietary antioxidants reduce the risk of CVD. Now, antioxidants are good at removing free radicals. These free radicals cause oxidative stress. In this case, They will cause damage to tissues, and if that damage occurs in the blood vessels, there is a risk of formation of plaque due to cholesterol building up around the damaged area within the blood vessel. So let's continue down here. They say, devise a study to confirm that antioxidants reduce the risk of CVD. If they ask you to devise a study, you have to make sure the study is valid. You have to work with large numbers of people to ensure that we have a lot of data to compare with. So use large numbers of people for the study, and they should all have non-known cardiovascular condition because we are studying the risk of CVD. If the condition is already there, then what are you trying to study? They should have the same risk factors, they should have similar lifestyles, and if possible, let them have similar non-lifestyle factors, like let them be of the same age, let them be of the same sex. Then to one group, we could give antioxidants, and to the other, we could give a placebo, meaning something that is not an antioxidant. And then both groups can be monitored for longer periods of time to see if there is progression of cardiovascular disease in order to see if the antioxidants are working or not. Let's continue. Here they say the coronary artery may become completely blocked in people with CVD. The graph shows how the energy released by heart muscle cells changes after the coronary artery becomes completely blocked. So we can see here vertically, there is energy released by the heart muscle, and horizontally, we see time after coronary arteries become completely blocked. They're going to say the heart muscle cells no longer contract 8 minutes after the coronary artery becomes completely blocked. Also, the heart muscle cells begin to die 20 minutes after the coronary artery becomes completely blocked. So moving to the next page, let's explain the effect on heart function after coronary artery become completely blocked. They want you to use the information shown in the graph and your own knowledge to support your answer. Here I began by looking at the theory of everything. I said there will be less blood supply to the heart muscle. This is if in the process of the blockage of the coronary artery, there will be less blood supply to the heart muscle. If that occurs, there will be less oxygen and glucose delivered. Therefore, there will be less aerobic respiration and less ATP will be produced. If this occurs, it means the heart muscle is now going to respire anaerobically using glycogen that is stored in the heart muscle because there is less oxygen coming to that area. So I said anaerobic respiration will then become the major source of ATP. However, this will produce lactic acid and this will decrease the pH around the heart tissues. Enzyme activity will slow down. This is going to occur due to pH changes, so it's going to slow down as the enzymes get denatured. Denaturation of these enzymes is due to lowering of the pH due to the increase in the lactic acid due to anaerobic respiration. So I said since glucose is not delivered anymore, glycogen will be used in vast amounts as a substrate for anaerobic respiration, but it's going to be producing less ATP. Based on the graph, energy released from the heart cells begins to decrease with time after blockage. I want to take you back to the graph. The heart muscle no longer contracts 8 minutes after the coronary artery is completely blocked. If I come at the 8-minute mark, I see the energy release is 53 AU. And when I come at the 20-minute mark where they say the muscle cells begin to die at 20 minutes, we can see it's between 23 to 24 AU, and that means the energy is really, really low. So when I take you back here, I said the heart muscle cells no longer contract after 8 minutes because the energy released, which is that, is not enough for the required muscle contraction. Then the heart muscle cells begin to die after 20 minutes because the energy is very low for cellular processes to maintain cell viability. This brings us to the end of question 7. Let's continue to question 8. Question 8. People with lactose intolerance cannot digest lactose. Lactose intolerance is due to a lack of the enzyme lactase, which are the products of lactose digestion. When lactose is digested by the enzyme lactase, we produce galactose and glucose, so the answer here should be a C. The next part says, explain how the three-dimensional structure of lactase affects the mechanism of action of this enzyme. when this enzyme is working, it produces an enzyme substrate complex as the substrate, in this case, which is lactose, binds onto the active sites. So if we are explaining the mechanism, we're going to think about how the enzyme substrate complex is going to be formed. So I said, and again, they want us to include a three-dimensional structure. So I said, lactase has hydrophilic R groups. The reason for saying this is because this is part of digestion, and in digestion, the enzyme is going to be soluble in some fluid. So lactase has hydrophilic R groups, so it is a soluble globular protein. It has active sites that are complementary to the shape of lactose. Lactose in this case is the substrate. Lactase and lactose collide, then they bind together, forming an enzyme substrate complex, which you see here. That leads to the lowering of the activation energy required for the reaction to occur or for digestion to occur. The key thing here is, as this enzyme works, it's going to be soluble. And because it's an enzyme and it's a three-dimensional structure, it is going to be a globular protein. This globular protein has hydrophilic groups or hydrophilic R groups that are going to make it soluble. During the reaction, the enzyme and the substrate collide. When they collide, they bind onto each other, forming an enzyme substrate complex. That enzyme substrate complex causes a reduction of the activation energy required for that reaction, so it occurs or digestion occurs at a lower temperature. So let's continue. Here they say people with lactose intolerance can drink lactose-free milk. Lactose-free milk is produced by treating milk with lactase. There are two ways of removing lactose from the milk. The first one could be mixing a solution of lactase with a milk-free lactase, enclosing the lactase inside permeable beads, and pouring the milk over them or immobilized lactase. The diagram shows these two methods. So we have this one here. We have milk and lactase in the same, this is a free lactase. And we have this one where the lactase is not free, but it's going to be immobilized inside permeable beads and the milk is poured onto it. In this case, we have lactose-free milk as well. So the first question says, suggest one advantage of using immobilized lactase. If the enzyme is immobilized, it means it's not going to be present in the milk, so contamination of the milk is going to be avoided by the enzyme. Number two, if the enzyme is immobilized, it means we can retain it and it can be used for continuous processes in order to digest more milk in the future. So I said the produced milk will not contain lactase enzyme, in this case with immobilization. Also, immobilization allows for the reuse of the enzyme. Let us continue. The table shows the effect of pH on the activity of free lactase and immobilized lactase. So I wrote a lot of stuff on this page. You forgive me. I will explain with this part a little bit later. So they say, explain the effect of pH on the activity of these two enzymes. The key thing is, I want us to first see how these enzymes are working. For this one, as the pH increases, we see there is an increase in the activity of the enzyme until around this range. So we are not sure if the optimum is here or here, but we know between pH 4 to 6, there is going to be an optimum before it begins to go down. So moving from about 5 going downwards, we see the reactivity decreases. That is observed there. And here we can see it's about the same thing. So here I said from the data, decreasing pH, meaning from 5 downwards, decreases the enzyme activity in both methods. And the optimum pH for both is between 4 to 6. We cannot exactly tell. So if you want to find out the optimum pH, you have to redo the experiment between this range and try to see where the optimum lies. So I said, change in pH causes ionization of the R groups. Now, I did not use R groups in my demonstration of ionization, but I used normal, like the amino acid, non-R groups. Let's assume we have this amino acid. This is the basic structure of the amino acid. If we put this amino acid in an acid, it means the nitrogen is going to get another hydrogen, which is the H plus from the acid is going to be gained here, and this is going to be an ionized amino acid. In alkaline as well, meaning pH has changed, the hydrogen from here is going to be stolen away to interact with the OH from the alkali in order to form water, Again, this is going to be ionized. So changing pH can cause ionization, whether you're ionizing due to addition of a hydrogen here or you're ionizing due to the removal of a hydrogen here. The ionization I'm trying to talk about should be in the R groups, not in the main part of the amino acid. Remember, R groups can also have carboxylic parts, and sometimes they can have amine parts. So I just use this as an easy example for you guys to understand what I'm talking about. So again, to emphasize, change in pH causes ionization of the R groups. Some bonds can break, causing the shape of the active sites to be changed. In this case, once the R groups are ionized, some hydrogen bonds are going to be broken in specific areas, or some bonds are going to break, and when they break, it means the structure of the protein is going to be altered. So I said, based on the table, the immobilized lactase is active in a wider range of pH because we see it has greater activity. Even when the pH goes higher, there is a wider range, and when the pH was lower, there was more activity. So it has a wider range of pH in which it can work. Finally, I say this is because immobilization ensures that those R groups are held in position where the active site does not easily change shape. Here, this is an extra explanation about the ionization. I said ionization of R groups in acidic or alkaline pH can alter the position of the hydrogen bonds and thus alter the shape of the resultant protein. This affects the active sites, meaning it causes denaturation. So let's continue. Here they suggest how the rate of activity of the lactase could be measured. They want you to include appropriate units. Since we want to measure the rate of activity of an enzyme, we need to know the change in concentration of lactose within a specific time. If you use concentration, you do mole per decimeter cubed per second, mole per decimeter cubed per minute. So I said rate is change in concentration of lactose over time. So you measure the change in the concentration of lactose. That concentration is in mole per decimeter cubed, and you divide that by the time taken in seconds for the concentration to change. So in this case, my units will be mole per decimeter cubed. Down here they say congenital lactose intolerance, CLI, is an extremely rare genetic disorder. Most people with CLI are found in one country. Suggest why most people with CLI are found in one country. If there is a genetic disorder that is in one region, it means it's a very recent mutation. If that mutation has occurred in an individual or individuals, they could have had children, but the children and their parents have not left that region, so it has not spread to other areas of the world. So I said, because the mutation occurred in the gene of the people living in that country, the people with the mutation had children, but they have not left the country. So the mutation is new and it has not been spread to other countries. That is the only reason as to why it could be present in only one area. So this brings us to the end of question 8 as well as the end to this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.